Welcome back for the third session in this fascinating conference on deuterium depletion. And I think I can safely say that our next speaker, Professor László Boros from California, is no stranger to this part of the world, and also has probably, although Hungarian by descent, has almost certainly traveled the furthest to be here. It's a very great pleasure to meet him. I've known his work for a little while, and I'm very much looking forward to hearing what he's got to say on his most recent developments on 13C tracer glucose-based metabolic profiling in relation to deuterium depletion. So, of course, you'll be pleased to know you're not pressed for time. We've only got the two speakers in this session, unfortunately, because Dr. Gabor has not been able to make it. So, over to you.
Good afternoon, everyone, and thank you very much for the opportunity that we can talk about the new models that we have built in connection with previous old data or data that we have generated previously. And you have a question? No? And that would be too early in the talk. But anyhow, so as you all know, we are using 13C tracer labeled substrates, and glucose is the one that we used in this particular study, and then we built additional models that you have heard before. So, but for the talk today, I figured I would actually give you an oversight of how glucose metabolism coming into the field now as part of systems biology, and what we found in deuterium-depleted growth environments using pancreatic cancer cell lines. And I think there's a need to cover some of the metabolomics developments that are taking place in the drug development industry, and I'm going to tell you why. Actually, there are great applications just as it comes down to deuterium depletion or any alternative, any new drug or any new cancer treatment modality, because actually the cancer field and in general drug companies are not doing that well based on the unsuccessful events in genomics research. I mean, it's truly a disaster, but I'll talk to you a little bit more in details. So the main problem in cancer, and I'm just going to talk about cancer in this part of my talk, is that up to date, there are about 400 oncogenes. Those are in the literature. You can just type in oncogene and you're going to find a bunch of them. And besides of that, there are about 300 tumor suppressor genes. So when you actually start playing with these scenarios, and if you have interest in generating permutations, what that would yield as far as drug development efforts, you can kind of see very early that indeed this is just a huge task as far as developing drugs against these oncogenes or tumor suppressor genes. Now, besides of that, there are about 15,000 cancer-related mutations observed and reported in the medical literature, which is a pretty huge task when there's a drug developer who wants to target any of these. And obviously, there are no efficacy issues yet related, and most of the oncogene stories and most of the tumor suppressor stories are, at best, are just confusing. I mean, I've seen confused scientists that, you know, they would be just confused whatsoever. And I'll tell you why and where the confusion comes from. Now, for example, KRAS, which was the biggest, that was the biggest, I would say, in gastrointestinal cancers. KRAS and KRAS mutations were really entering the field very early as a typical oncogene. Now they have to actually separate two kinds of KRAS. One is which has an oncogenic mutation, and the other one that has mutation, but it's not really oncogenic. So, I mean, I'm a little less confused because I don't really go into this in details. I just kind of acknowledge the disasters that the genomic research generates these days. And truly, with all the respect, I really have never been interested in exploring any of these any other way other than to provide some functional metabolomics type of data that cover some of the findings or some of the functional impacts that are carried by these oncogenes. And I'm going to show you some new developments that I think we are getting successful at finding out really which one to pick for drug development. Yet, even the drugs that have been introduced to the market as, let's say, magic bullets or the typical oncogenic drugs that are targeting, for example, kinases, they are really clinically, they are not that efficient. And we have studied, for example, Gleevec, which was the magic bullet in leukemia treatment, which obviously has some clinical benefits, but as far as the lifespan or disease-free survival, it's really not that impactful as if you compare with the cost it costs. It's about $20,000 in the US, just 1 year of supply. So it's not everybody can afford it, obviously. And gene therapies actually have been discontinued in the US because they killed this young kid in Pennsylvania with a known metabolic disease when the gene was clearly known. It's a cystic fibrosis type of disease, and they used an adenovirus as a vector, and they cured this kid. So all the genetic or gene therapies in the US are now suspended, and they have been suspended for about 7 or 8 years because it's just really unpredictable what a gene will do once you transfer it into a human host. So as the story develops, that now, and I know because I work with the Food and Drug Administration, over 95% of new clinical trials or drugs that are actually going through clinical trials are going to fail. And they fail because they just don't pass the phase 2, phase 3 trials, which are actually randomized or obviously blinded studies. So it's really a not very promising statistics. Now, besides that, and this includes all disease areas, I think this is close to 99% as we speak today, the rate of failing drugs. And the pharmaceutical companies are actually going back to make generic drugs. Eli Lilly, which is one of the largest pharmaceutical companies, now competing with generic drug makers because they don't really have new drugs. They don't. And besides that, actually, they are disassembling manufacturing plants. For example, the plant where they were making Viagra, they disassembled it because Pfizer is not going to make Viagra anymore, and they don't need that facility anymore because they don't really have new drugs. So they cut down manufacturing operations, and because of that, they start firing their scientists because they don't really have costs or they don't have revenues to support much activities in the research field because they don't manufacture drugs. So they don't have actually proprietary drugs, meaning that they're not going to have sales, huge drug sales in the US. And on top of that, because of the failing clinical trials, there are unprecedented financial burdens that these pharmaceutical companies and their executives have to make, simply because the new drugs are actually just not as efficient, and they fail phase 3 clinical trials. For example, one compound, take it from the laboratory or the academic side to a drug, to a marketed drug, it costs about $9 billion. I mean, it's really an expensive operation, and failing at the end of that process and the phase 3 huge clinical study phase, it's really a waste of money. Or it's really just a huge burden on them. And let's say the best example is this angiogenesis kind of hysteria in the US, which started about 15, 20 years ago. And, anyone who thinks realistically about how cancers grow, what is their basic biochemical pattern, they know that angiogenesis may be useful in some cancer treatments, but it's really not true of every cancer, is that they really depend on this process. So be it, Roche just spent $48 billion because their angiogenesis drug did not pass the phase 3 clinical trial. $48 billion. Even for a huge pharmaceutical companies like Roche, when they have to buy up operations from Genentech, this is obviously public, so I'm not telling any kind of confidential information. You can check this on the internet. Because of the mammalian cancer drug failures in the US, they lost about $48 billion. So because of these costs of these failing drug trials, and there are no taxable pharmaceutical revenues or income in the US, ventures or investors or private funders or people who are interested in funding research are just running out of money. Why? Because they invested in here already and no return, meaning that they can't really invest in new opportunities or drug opportunities. And besides of that, there's another problem, and that is we now have molecular biologists everywhere, meaning that we have only and almost just molecular biologists working here and there who don't really know how to interpret any other kind of data than more of like gene expression data or signal pattern or signal patterning data. And when I tell them, listen, can we talk about something else? They can't. So obviously, because of this very narrow training in genomics, and there's a generation of scientists now who does genomics and nothing else, they are further emptying the pharmaceutical pipelines, meaning that in the next 15 years, probably there's going to be no new drug in the US market, because that's how pharmaceutical companies' pipelines are emptied. So, and this is statistics, so we know these trends are occurring, and it's really truly just an alarming trend. So when it comes to deuterium-depleted water, for example, and its pharmaceutical applications, I think truly there are obviously steps involved, but if it's effective, if it's efficient, and there's a mechanism that we can link with treatment, with deuterium depletion treatment, those biomarkers can be identified from plasma, or we can actually find patients who are in a certain metabolic phenotype that is responsive to deuterium-depleted water. We could design very successful clinical trials, obviously, and we actually have some very good ideas how those could be guided. So this is if you talk to an oncologist or you talk to a molecular biologist, this is what they're gonna tell you. There are these oncogenes, they get activated. There are these tumor suppressor genes, they get lost, and this is how cancer develops. You see all these arrows are going around, and they develop drugs. Again, these and then when they use these tumor suppressors and oncogenes in the different models, they switch back, meaning that the tumor suppressor becomes an oncogene, and oncogene becomes tumor suppressor gene. So usually when I talk to my colleagues at UCLA who do signaling research or genomic research, I usually tell them that this is just not going to be effective whatsoever. This is just not going to be efficient. So truly, to understand what is behind these oncogenic activation or tumor suppressor inactivation processes, and what is the metabolic fingerprint behind these reactions or processes. Actually, if there are oncogenes, I don't think there are oncogenes. I mean, there are genes that are involved in certain stage of the disease, but this oncogene baloney is just, to me, it's just really, truly out of style. I mean, this is just truly, based on the continued failure of these drug targets, it's just really truly, it's actually getting out of, you know, the vocabulary now in the US at meetings. And the same is true with tumor suppressor genes. So what we have known for a very long time is that actually there is a metabolic process which is linked with tumor growth or cellular proliferation or uncontrolled cell proliferation, and that is what we call the Warburg effect. He was a German scientist, and actually he found that actually tumor cells use a lot of glucose and they produce a lot of lactate. So if cells engage in this glucose to lactate transfer through pyruvate, and they consume a certain rate of glucose and they produce a certain rate of lactate, we identify those cells as Warburg type tumor cells, and we know there's metabolically something is really inefficient in these cells, and many believe that actually glucose is used for lactate production and energy production, because actually tumor cells would gain 2 ATPs, net ATP surplus from the process of glucose to lactate conversion. And indeed, yeah, just my friends just came from my hometown, so hi. Take a seat. So, because this is a big event, trust me. So, and in the process there is signaling pathways involved, but really truly the main process or the hallmark of the disease is this glucose to lactate conversion. Now, when all these oncogenes get activated, besides lactate production, which we know precisely how it occurs, all the other molecules that are involved in cellular proliferation, they start falling from the sky. This is what molecular biologists think, at least. This is their understanding of cancer. So as the birds are flying, there's a purine base coming from somewhere. Actually, there's the deoxyribose produced. So if you ask a molecular biologist that, what do you think, besides lactate, what else is happening in tumor cells? You ask them specifically, what do you think, how is ribose generated in tumor cells? They would just look at you like, it's probably coming from the sky. I mean, if you don't know where it's coming from, usually that's the answer. So actually, to synthesize ribose in tumor cells, there are specific pathways. Actually, there are actually very specific reactions which are different from normal cells, which produce ribose in tumor cells. And knowing those reactions historically took the scientific field back to some old papers that we published back in the 1990s, which I'm going to cover for you and see how molecular biology is trying to recover information that is really, they should have done it in the first place. So when it comes to understanding everything else besides lactate production, the best understanding is just they are just available, or the which is pretty funny, but that's how they think. So, and when I talk to my colleagues at UCLA, and they happen to be molecular biologists, I ask them, "Hey, Steve, or whatever, what do you think? How the cell proliferation process is accomplished, as in particular ribose and deoxyribose synthesis?" And they just say, "Well, those are there." I'm like, "Yeah, sure." Those are there. But anyways, so back in 2000, in the paper Genes and Developments back in 2010, Dr. van der Heijden from the Massachusetts Institute of Technology, which is in Cambridge, Massachusetts, which is a pretty prestigious research institute, they are the ones who put men on the moon. So, and I told this to my colleagues at Stanford Research Institute, because I work with them closely. And I told them, they cited me by MIT, and they are the ones who put people on the moon, and the Stanford guy said, no, we put people on the moon. I'm like, okay. I don't want to get into those details, but it seems that from the Massachusetts Institute of Technology, this group is really interested in metabolism and explore how metabolism affects cell signaling. And clearly the truth is, when metabolism strikes back, that's a major turnaround in the thinking of how oncogenesis occurs. And truly what they have been discussing is that there are metabolic pathways, and these pathways provide substrates for post-translational modifications, and those influence cell signaling patterns. So that's clear. I mean, metabolism has to be involved in the signaling process simply because they modify proteins. UDP glucose, which is one of these substrates that can glycosylate proteins, is the product of glucose activation. So obviously, metabolism and glucose transport has to be involved in cell growth other than just lactate production. And glucose, glutamine, and acetyl coenzyme A are all required for glycosylation, for example. And in turn, when there's limited availability of glucose or other substrates for cell growth, this whole system will shut down. So this supports two ideas. One is that these substrates are in fact important in regulating cell signaling through secondary modifications, and indeed they supply these building blocks for RNA DNA synthesis, which is actually the base process, or one of the crucial processes in unlimited cell proliferation. What the specifics of those reactions are is coming from, or at least going back historically, again, from the Massachusetts Institute of Technology by Dr. Stephanopoulos in the Journal of Biotechnology, where they actually review the field, and they cite one of the papers that we have published in 2003 in Blood, because, and this was related to a disease process, which I'm going to cover with some specifics, but yet they actually spell it out that they are able to quantitate and validate the effectiveness of some of the 13C tracers, and these are substrates that the cells are eating. These are the ones that we are using are glucose tracers, and as I usually just tell my students, those molecules are exactly the same as the sugar or the glucose that you would put in your coffee. It tastes the same, it smells the same, it has the same taste, it's all the same. It's more expensive. Your coffee is going to be more expensive, but everything else is the same. If you can afford it, go ahead and do it, but usually it's $400 a gram, so it's not really a wise idea. However, those are actually chemically identical to the glucose that the tumor cells are using. And when it comes to discussing what they can actually now study with these 13C tracers, they are pointing out that there are scoring algorithms that are employed to determine the optimal tracer concentrations, and by then you can actually study glycolysis, the pentose phosphate pathways, and the tricarboxylic acid cycle. Indeed, there are already applications of these processes back in the medical literature, one from 2003. And now they consider this application as one of the gold standards in clinical research. So in another paper, which is also just in the discussion, what they say is that now they can actually perform metabolic flux analysis, and actually the most powerful and the most meaningful applications are coming from us, first in 2003 and then from another team in 2007. So there are many, many applications. There are much of the flux analysis are through measuring metabolite concentrations on the large scale. But if you use a tracer, if you actually trace those metabolites, you can actually generate the most powerful and the most meaningful applications. So I called Dr. Stephanopoulos after this paper. I told him, listen, George, this is really nice, because you actually spell out that what is the use of the tracer-based technologies, and historically where it goes back, the first efficient clinical applications. So, and that paper, which they are citing now, goes back to November 2003. It was published in Blood, and it was in the hematopoiesis section, and this was actually describing a disease process which has been first identified in 1969, and for 30 years nobody knew what was causing the disease. Actually, it was a very rare type of anemia in babies or kids. And actually they responded to thiamine, which is vitamin B1, and nobody really understood what the process was. So with the Harvard team, with the Boston Harvard team, we sent in our papers where we clarified exactly what is the metabolic abnormality in these kids, in their fibroblasts. And we got this very nice editorial by Dr. Rolf Green, who is at UC Davis. I don't know him in person, but he is also affiliated with the UC system. He was likely one of the reviewers of the paper, of this paper. And in his editorial, which came out with our paper, he actually stated that we actually identified, and we looked at inaccessible, we actually identified gene function analysis, and we actually reached into the cell which were actually inaccessible in the metabolic maze. And then actually we provided a key to better understand changes in substrate flow as basis for drug mechanism and disease characterization, and we actually pinpointed the metabolic abnormality that was the underlying process that caused this disease. And this was a very nice editorial by Dr. Green, but really truly it's been, to its clinical value, it's been sitting for almost 10 years now in the medical literature, until actually we got cited in Nature back in October, where they actually cite another paper of us which goes back to 1998. And in Nature Reviews, they actually spell out for some of the applications, they spell out why they cited that paper, and this is what they wrote. This study, which we published at Ohio State in Medical Hypothesis in 1998, some 13 years ago or 15 years ago, this study was among the first modern studies to track carbon in cancer cells and called into question common assumptions about cancer metabolism. So, but the molecular biologists would say, like, those metabolites are falling from the sky. We were actually identifying those biochemical reactions which contributed to tumor cell proliferation, and this is what they are citing here, or this is what they are describing here. Because truly, when they actually review the literature, there are already very important applications that need to be considered. Okay, so this is the paper they are citing, and in this paper in Medical Hypothesis back in 1998, now a scientist, what would you do when you have no funding? You write your proposals to the NIH, the pink sheet comes back like those people have no idea what they are reading or looking at. They give you some kind of, or try to give you some kind of review which you wouldn't even understand. It's like they are so far off, I would say. They have no clues of what they are actually reading or what they are reviewing. So you just kind of sit there and you say, like, why would I bother with these people? What can they do as far as reviewing this grant proposal? So as you have no money, then you publish in Medical Hypothesis because you have an idea, I guess, but without money, I mean, it's hard to, go anywhere else, but you cite it. And you write your paper there, and with your co-workers at Ohio State University. By the way, by my chair at that time, Dr. Allison was the son of Edmund Allison, who was one of the authors in the Zollinger-Ellison syndrome, the gastrin-producing pancreatic cancer that causes ulcer. So historically it was a very interesting work. And what we wrote in this paper that actually glucose metabolism, and we only found bacteria in the literature, but we described in this paper that actually glucose metabolism related enzymes sit on certain, are activated by certain transcriptional factors or operon segments, and they are actually clustered. So one genetic event may cause a chain of metabolic events which are linked together in a certain pattern that actually they would promote cell proliferation, one way or another. And some of those clusters include some key enzymes of the pentose cycle, fructose cytoheptulase, the biphosphatase, phosphofructokinase, transkylase, glyceraldehyde 3-phosphate dehydrogenase. These are enzymes which are involved in ribosynthesis or nucleic acid synthesis, and eventually we describe in this paper that if these clusters are active in cancers, you can actually, with one single genetic event, you can activate a whole range of metabolic reactions if they are clustered, just like in bacteria, which actually will generate some cancers could therefore be developed due to metabolic changes within the cells and the host, which are marked by defective regulation of glucose metabolism as clustered together in genetic events. And to understand such strategies could bring new treatment to the horizon. So, and this was 13 years ago. And truly, when it comes to some applications, and now this is, I think our papers are cited because of this section. It actually tells you something that was not seen at that time, that one signaling pathway, you don't have to look for another signaling pathway that signals for another signaling pathway. When you see a signaling pathway, you should probably see what enzymes get activated, because that's the whole name of the game. Now, how many signaling pathways it takes to reach there, it doesn't matter. It's really not that the essence of the game. It's, if you talk to a molecular biologist, their thinking is pretty much that, let's say somebody sends you a package, a FedEx package or a DHL package, and by the FedEx guy pushing the doorbell, the package was delivered. That's not true. He signaled, and if somebody's in the house, yeah, indeed the package will be delivered, but the signaling itself does not mean that a biological event occurred. In fact, more signaling means that nobody's home. So molecular biologists, about 50% of the time, they actually draw the wrong conclusion. Based on the signaling pertains, yet there is no biological response. And I've seen all those scenarios coming along. So because of this, we started actually applying these models very efficiently, because in the Public Library of Science, very recently by my colleague at the University of Pittsburgh, they just discovered a energy-producing pathway for tumor cells that is related to only one genetic event, and that's a c-Myc activation. And this gene was actually first discovered in Burkitt's lymphoma patients, where cancer cells show chromosomal translocations, and this is associated with chromosome 8, and it's frequently involved in the disease process. And cloning the breakpoint, this fusion chromosome, revealed that there was a similarity to a viral oncogene or a virus causing tumor, which they called v-Myc. But indeed, when we actually we activate this one c-Myc gene in these tumor cells, what you're going to see is that there's going to be metabolic enzymes which are just based on one genetic event in the active tumor cells, which actually have this c-Myc mutation. Actually, the control tissues, which are just adjacent tissues, or the short-term application of c-Myc will not cause any metabolic adaptation. After a certain period of time, those metabolic enzymes get activated, and this is the list what those are. And once you withdraw the c-Myc, those enzymes get actually withdrawn as well. So truly, this one single metabolic event, yeah, and in the meantime, it also signals to PCAT and MCAT and MNM, and you know, I've seen c-Myc upregulation, downregulation signals and genes in the literature in the last some 20 years. They have been studying c-Myc, and I've seen mutations, and I've seen all kind of up and down-regulated signal transduction pathways. But as far as metabolic pathways go, this is the first when we identified what those enzymes are. And sure enough, if those enzymes get activated, these tumor cells will become different than even the tumor cells that are the Warburg type. Why? Because what they do is they, from glycolysis, they take in phosphoglycerate, which is glyceraldehyde. They actually convert it into serine. From the serine, they produce glycine. And while they produce, from serine, they produce glycine, they start running this ATP synthesizing loop, which will yield them 3 extra ATPs compared to the Warburg effect, which will only give you 2 ATPs. So indeed, tumor cells, little by little, but they can modify their metabolic reactions or metabolic pathways based on one single genetic event, and that activates a chain of reactions or chain of enzymatic reactions, which actually will give them extra ATP if they oxidize this glycine completely down to CO2 based on the number of carbons that are involved in glycine. We know there's 3 carbons in there. They will be actually serine and glycine conversion into CO2. 3, eventually you can oxidize 3 carbons, so you're going to generate 2 extra or excess ATPs that you would be able to generate with lactate. So obviously this is a metabolic chain of reactions which can be targeted by drugs for further development. And actually when we correlate tumor cells and their malignant transformation, this pathway seems to be a very important key player in the process. So it started from a gene array. We're not against gene arrays, but we just want to use them in a more targeted pattern, meaning that we are more interested in what is the metabolic impact of these gene modifications. And I just, for clarity, I just wanted to, the expression profiles of the genes of the array that we saw was a c-Myc, doxycycline-inducible c-Myc transfection, and actually it was liver cells were transfected with it, and controls are the adjacent normal tissue. The pre-tumors are the 4 weeks with the c-Myc. The tumors, the true tumors that have picked up proliferation are 8 and 10 weeks of c-Myc, and regression of tumor samples were 3 days after removal of the c-Myc, and this is where they reverted back to the normal gene expression profile of those metabolic genes. So indeed, there are applications of those very old, or more than 13 years ideas, and that is practically when tumor cells respond to a signal, growth signal, they activate based on clustered metabolic events, they activate certain metabolic enzymes, and those enzymes can be targeted for drug treatment. So, and now, after kind of giving you a historical overview, I'd like to go to the deuterium depletion story, because I think that's another very interesting link as far as how it can be used as a cancer treatment modality, and where is the marker value, where is the edge where we can, or the people who apply deuterium depleted water, where they can actually apply it best for the clinicians. So again, this is going back to, and Dr. Jenks was editing this book back and forth back from the 1950s, but there were several editions back in 1970s, and this is not new to anyone of us here, that actually the rate of an enzymatic reaction in which deuterium is being transferred is several folds slower than that of the corresponding reaction in which hydrogen is transferred. So this means that actually if you, again, just review it with you again, if you replace deuterium with hydrogen, some metabolic reactions will definitely change kinetics and speed up or accelerate the rate. And obviously, one of the most critical enzymes that we are interested in studying is, and it's a very important, becoming a very important drug target in cancer, is this citrate condensing enzyme. They called it citrate condensing enzyme back in the 50s, but now they call it citrate synthase. And depending on water or deuterium oxide environment, actually there's going to be a decrease or increase in the enzyme's flux to produce citrate, and that's practically what you can see. And this is historic data. This goes back to the 1960s. And this shows that actually deuterium-depleted water increases kinetics of the enzyme several-fold, at least twofold, when you compare it with the deuterium-labeled water. So obviously, when it comes to citrate synthesis, which supplies the TC cycle, for example, and this goes back with your research. So if you produce citrate, you have to kind of consume it one way or another, you have to use it one way or another, so it just can't just kind of hang around in the mitochondria, because mitochondria has a ATP energy producing chain of reactions. So once citrate synthesis is adapting to the H2O environment, this would be a very important hallmark of really truly what the fatty acid synthesis or mitochondrial activity would do. And this study, for example, this paper goes back to the 1960s, and obviously just citrate synthase now previously was used the citrate condensing enzyme, but it has a great response to deuterium depletion. Now the question is, and this is truly what the question is, how can we measure this enzyme, for example, in a living cell? Because to do it in test tubes, it's maybe easier, but actually move to a living organism or moving to human beings, this might be a challenge, and this is why we have developed, and we are collaborating with Gabor, because this is truly a very important biomarker to measure fatty acid synthesis, or the rate of fatty acid synthesis in tumor cells, and also the effect of this on deuterium-depleted water. So you have to use glucose, and I'm sorry I have to go back to my little glucose molecule, because this is actually a 6-carbon glucose molecule. The animation was done by my nephew, who is in the audience. I put his doctor in a little bracket because he's graduating from medical school in July. So he's not a full doctor yet, but he's pretty much there. So let's give him some credit. So, Tamás, stand up for a second. That's where he is. So that's where he is. But anyways, the way we actually perform these studies, we mix this glucose, which has 2 heavy carbons on the 1 and 2nd position, 90 mg%, with naturally labeled glucose in the medium. And what we do is we follow the trace of this glucose in and throughout metabolism. And this is where citrate will be synthesized, and I'll go back to that process. But let's stop here, because this is where the c-Myc-induced metabolic single carbon metabolism starts out. This glycerol is taken out for the which I have talked about. This is another oxidation pattern for glycerol, and yet in the mitochondria, citrate will be generated from acetyl-coenzyme A, and based on how much carbon would accumulate from glucose into, for example, citrate or fatty acids, we can actually measure this flux. We can now, in humans or in animal models or in cell culture, we can actually measure this flux and its response to deuterium depletion. So this very expensive sugar that you can put in your coffee, this can actually do something else, and that's worth of the money. That's worth buying the sugar for, because actually you can follow and trace these two carbons, and this is what the MIT is referring back, that when they are saying that we are able to trace these carbons, this is why they cite our papers, because that's what we have been doing. And this is obviously one of the key steps when metabolism commit to generate citrate from pyruvate. Pyruvate can be used for lactate production. Pyruvate can be used through the American. So there's a number of uses for pyruvate, but when pyruvate enters the TCA cycle, then there's going to be a commitment that citrate is formed, and the citrate synthase process is an irreversible reaction, meaning that citrate cannot go back to anything else. It has to be metabolized further, which I'm going to show you, or it has to be shuttled from the cycle. And this is the citrate synthase reaction, and what can happen to it, it's from the mitochondria, it can be shuttled, and citrate is accumulating in the cytoplasma. So when citrate accumulation comes in the cytoplasma, obviously there are a number of scenarios that can happen to citrate, and one of them is actually the membrane synthesis process, or fatty acid synthesis, or fatty acid synthase committed reactions where actually citrate is consumed up, or at least two carbons from citrate, acetyl coenzyme A, is actually becoming part of the fatty acid synthesis chain and fatty acid synthesis process. And this is where the citrate shuttle, measurement of citrate shuttling becomes very handy when it comes to deuterium-depleted water research, because we know from historic data that actually this enzyme responds to deuterium depletion, which means that actually it increases citrate production. And I'm going to show you some data that we have generated in collaboration with Dr. Chaumier. So citrate can be shuttled and actually can be used for alpha-ketoglutarate or glutamate synthesis. This is what we call the cytosolic NADPH production process. This is actually also an isocitrate, citrate, isocitrate dependent process. Instead of NADH, NADPH is produced in the cytosol, but the result of glutamate synthesis. And glutamate can also be synthesized in the mitochondria, and the result of the mitochondrial alpha-ketoglutarate production is also glutamate labeling. And if we use 50% tracer labeled glucose after 24 hours, and this is the deuterium depletion in pancreatic tumor cells after 24 hours, what you can see is the labeled fraction is about 23% in all cultures. So it seems that neither the positional labeling nor the labeled fraction in glutamate after deuterium depletion, or this level of deuterium depletion in this tumor cell model, is not a major responding flux to deuterium depletion. So it seems that oxaloacetate to citrate, citrate to isocitrate, isocitrate to alpha-ketoglutarate, and glutamate production goes just at the rate as the control cells. However, when we look at the fatty acid panel, in addition to the glutamate synthesis panel, when we look at the fatty acid synthesis panel, we're going to see some changes. And obviously, the best way when we come to isotopomer data in response to any drug treatment, the best way of reviewing this data is to find correlation patterns between the deuterium depletion or level of deuterium depletion and some of these fluxes, which include glucose deriving or 13C labeling in, for example, myristic acid. This is the 14 carbon chain length short chain fatty acid, palmitic acid, which is the product of fatty acid synthase, and then intracellular stearate, oleate, arachidic acid, behenic acid, and lignoceric acid. And these models, when you actually look at these labeled fractions and positional labeling, as well as their acetyl coenzyme A enrichment from glucose deriving acetyl coenzyme A, you can actually see the biological pattern, how citrate and fatty acid synthase respond. Do I have to finish at 1 hour, or can we finish? We have time, as I understand. Okay, so because this metabolic matrix data is a little bit complicated, it's hard to like compare like number by number and then just go and have coffee and come back. We don't know where we were, so it's just kind of it's hard to review this type of data. This is why with the Food and Drug Administration we developed a method where we actually tested drugs already using this what we call a regression statistics, or regression or phase plane analysis, where we actually, instead of looking at number by number, we look at regression statistics. And actually this is used now by the Food and Drug Administration. So with this type of data, we can go in and get some filing with the deuterium-depleted water when the time comes. But what we do in the flux analysis and the regression type of analysis data with the Food and Drug Administration is that we look at glucose metabolism at certain time points and how glucose is distributed into fatty acids, for example. In this case, it was cholesterol. And we measure the acetyl coenzyme A enrichment in plasma, circulating plasma cholesterol, and we determine how efficiently glucose, which is here in the it's the glucose curve of valproic acid, meaning that when we actually load glucose in these animals, glucose has a certain kinetics of losing its tracer, yet in the meantime valproic acid treatment decreases labeling into cholesterol fatty acid, meaning that something is in the liver gets disrupted, because the correlation statistics or the regression statistics between glucose labeled between glucose and cholesterol, which is dependent on HMG coenzyme A, and also fatty acids behave the same way. This is disrupted, so there is a low correlation which indicates drug toxicity and falling apart type of fluxes. And when we generate the same type of regression analysis for fatty acids obtained with decreasing deuterium concentration yet increasing level of deuterium depletion, then we see really just very interesting findings here. And I like to kind of go through with you because it's really interesting. So we were talking about glutamate because glutamate is the product of the TC cycle. We've been talking about glutamate. Actually, there's no correlation between deuterium depletion and glutamate. Well, yeah, the correlation, the R square is about 0.3, and the positional labeling is really not very responsive to deuterium depletion. But when you look at, for example, fatty acid synthase flux or labeling of palmitate from glucose in these tumor cells, there's a relatively good correlation, but there's a negative regression between increasing deuterium depletion and actually fatty acid synthesis. So actually, when you increase deuterium labeling decrease deuterium labeling water in these tumor cells, fatty acid synthase or fatty acid exchange or fatty acid labeling will not be as efficient as in your controls. And this is a pretty good negative regression analysis result, meaning that there are better ones, but actually this is pretty strong as far as negative correlation between deuterium availability and de novo fatty acid synthesis. And this is very important because we talked about this data yesterday, that fatty acid synthase, this is the product of fatty acid synthase. So indeed, it seems that this enzyme, fatty acid synthase, is a very efficient deuterium depletion responder as far as this tumor cell model goes. Now, there's another very interesting labeling pattern, and this is actually, if you look at All the fatty acids, I'd like to point out that all the fatty acids, as far as their 13C labeled fractions go, they actually have a negative correlation with glucose, except for one guy, which is oleic acid. And now I'm going to talk about this because this is really significant and explains a lot of findings that Gabor told me confidentially in the car yesterday, but I'm going to just tell you now because, you know, I'm not getting paid for my confidential, but no, it's just, I'm just kidding. My Chinese colleague was there too, so he heard everything. So I'm just joking. Yet, when you look at the acetylcoenzyme enrichment for stearic acid, and when you look at the acetylcoenzyme enrichment for oleic acid, you see a very positive correlation. And I mean, would you know why?
Yeah, of course.
Yeah, Gabor would know. Yet, let me tell you. It's actually how cells transport fatty acids in the form of high-density lipoproteins. The high-density lipoproteins would not be able to assemble unless there is a certain portion of oleate and a certain portion of stearate provided in the triglyceride, which eventually will condense with the high-density lipoprotein. So to be able to transport fatty acids, you actually have to increase labeling specific acetylcoenzyme A enrichment towards those fatty acids, and this is what these cells do. And these are actually gastrointestinal cells, so the same finding is expected if you want to use this as a biomarker for fatty acid transport. The other very interesting finding there is, besides decreasing palmitate turnover, the other very interesting finding is here, is it's related to behenic acid and lignoceric acid. And again, I think this is the more important guy. Anybody knows why lignoceric acid is important in membranes? I'll tell you. It's actually the fatty acid of the nuclear membrane, because it's a saturated long-chain fatty acid, and the only strong enough fatty acid or rigid enough fatty acid to hold chromatin or DNA. So this fatty acid, actually this lignoceric acid, makes up about 30% of the nuclear membrane. So this is why it's so important, because it seems that deuterium-depleted water decreases glucose to lignoserate turnover, meaning that these cells are in a cycle arrest. They are not going to be able to turn their nuclear membrane over, and this you can see clearly from the lignoserate labeling pattern. While they are actually transporting fatty acids, stearic acid and oleic acid get labeled. In the meantime, they don't really turn over their nuclear membrane. So there's a difference of the type of fatty acids they synthesize, the type of fatty acid they label, and thus there's a major change or difference in their biochemical fingerprints and indeed in their phenotypes. So truly, looking at these models and looking at these fatty acid labeling patterns from glucose and using the regression statistics, you can actually see what is happening in these tumor cells. So indeed, if the patient comes in with a fatty acid synthesis type cancer, and we can actually tell that this is actually a cancer type where citrate gets shuttled out, we can get this patient treated, and we can measure its tumor response to deuterium depletion by looking at particular fatty acids that are involved in various processes: fatty acid transport, de novo fatty acid synthesis, chain shortening. You see there's not much chain shortening. So palmitate does not get really shortened very efficiently into myristic acid, which you would expect from a tumor cell. This is not a liver cell. This is not using peroxisomal beta oxidation very efficiently, but it uses chain elongation, chain desaturation, and also the long chain saturated fatty. And this correlation is really high. It's 0.099, meaning that it's really responsive to deuterium depletion. And you can actually now assign a value in a living organism real time or in a host, simply how efficient your concentration is working on these various types. So the next question I have to tell you is, so what's up about this fatty acid synthase? Well, actually we have a very good set of data or imaging patterns from La Jolla, which is in California, the Scripps Research Institute, where they actually characterize fatty acid conformations and fatty acid structure very efficiently. And I think this is also very important to understand that fatty acid synthase in mammalian cells and fatty acid synthase in fungus and bacteria are very different as far as their structure goes, as far as their kinetics goes, as far as their proteins go. They are two different enzymes. Now, while bacteria can actually grow in these prokaryotic organisms, which are bacteria, they can actually grow in deuterium-depleted environments because you can actually grow them in heavy water. Bacteria and fungus grow in heavy water. Mammalian cells, they can't. And tumor cells, it seems that actually this conformation of fatty acid synthase is very responsive to deuterium depletion, but this guy is not, really. And actually, I'm going to tell you this depends on these closed structures and these MPT carrier proteins, which are very important elements of this enzyme system. But anyways, so this is Mr. Fatty acid synthase with its specific conformation and reaction chambers, which are open just like really truly. And they actually have to have this open because they're actually chain length and the number of fatty acids that they store in there, the acyl carrier protein, with the acyl carrier protein, it's actually overwhelming in some membrane synthesis scenarios because these guys don't need membranes. They need a different kind of cellular membrane. But these guys are actually very dependent on a very fast turnover of membranes because they secrete insulin, they secrete neurotransmitters, they store and secrete other hormones. So these are more sophisticated cells. Yet their fatty acid synthase is more sensitive to deuterium depletion. So what are these subunits? You can find the acidic coenzyme transferase, which is actually this unit here in the bacterial. You can actually see this MPT or malonyl palmitoyl transferase, which you can see here in the bacterial guy. Actually, the largest proteins, excuse me, the largest proteins and the most important fluxes that are involved are these with these keto acyl reductase and synthase processes, which actually interfere or interact with the reaction chamber. And one of the characteristics of this enzyme is that it has an incredible sensitivity to pH, to point mutations, to deuterium depletion. It's such a big enzyme, it's like a Christmas tree, meaning that it has all these arms and flips. So if you eat the little sugar out from your Christmas tree wrapping, meaning that you replace the deuteriums with hydrogen, this conformation will change. So it's enough in this enzyme to introduce point mutations, and these are described what kind of flux change will occur. For example, this guy here, you see how big this chamber is, reaction chamber is in fatty acid synthase, and how different the reaction chamber is in these other fatty acid synthase conformations, and these are all result of point mutations. And here you can actually see what the flux-based modification is in response to any of these point mutations in proteins. And there are other, I think by now there are about 40 different conformations described in connection with this enzyme, with some significant loss of flux for processing one or another reaction involved in fatty acid synthesis. I'm not going to go through all of them, but actually you can review this in the literature. They are actually involved in various processes that are actually related to each of these subunits. And the bacterial guy is that it's just not going to change. It has no conformational sensitivity to nothing, practically. It's just such a solid protein structure that has no sensitivities to, excuse me, to deuterium depletion or point mutations. So, and I'm going to finish in a minute. Another important reaction that happens in the reaction chamber itself is this keto acid processing, malonyl coenzyme A and keto acid processing reaction in the chamber, which is actually uses water. So deuterium depletion in water and the deuterium that actually attacks, or the water that attacks and drops deuterium or hydrogen to this reaction chamber changes the kinetic dramatically. So this enzyme is not only sensitive to conformational changes in response to point mutations or deuterium depletion, but it's also responsive to various, to water itself and the deuterium that the water carries. Because actually the malonyl coenzyme A, the malonyl coenzyme A is attacked by water, and this reaction kinetics depends on water and deuterium depletion. So, so what would be the impact of such dramatic change in fatty acid synthase and citrate flux, increase in citrate flux? This guy, citrate that we are talked about, this guy, he's the main regulator and activator and regulator of fatty acid synthase, both activity and gene expression. Meaning that if deuterium-labeled water actually increases citrate production, which we know with low deuterium depletion, we can tell that with low deuterium depletion, you can tell in some of the fatty acids with the lower numbers, you can actually see increases in M2 labeling, meaning that indeed it seems, even though fatty acid turnover is decreased, actually citrate is pushing out. Even though the turnover rate is really not very efficient, your fatty acid, your citrate is enriched with M2 labeled glucose, which comes from acetyl-coenzyme A, and it shows an increase. So we know, indeed, and we can measure how efficiently deuterium depletion increases this this activation, this fatty acid activation process. The problem with fatty acid synthase is that it's a huge protein complex. So if you synthesize a lot of it, because citrate is carrying signals of that sort, what's going to happen is what we call is a system crowding, which means that these cells will eventually just choke whatsoever. All right, so thank you for your attention.
Thank you very much, Lazarus, for that real tour de force. It's wonderful to meet another metabolic biochemist that believes that metabolism is more important than genes.
You are?
Indeed. Always have been. Genes are merely there for metabolites to produce themselves. Yes?
I agree.
Anyway, enough of this facetious comments. What about any questions you'd like to put? Right at the back. We need a microphone back there.
I may have a comment and a question. The comment is that in plant kingdom, it's well known that in diseased plants, in many cases, the normal respiratory pathway, I mean from glycolysis and citrate cycle, etc., there is a shift into the pentose phosphate pathway. So we call it a shunt because it shortens the the respiratory pathways. And the reason for that, we think that it's the need and the production of NADPH, which is absolutely necessary for the enhanced biosynthesis in this kind of diseased plants. The question, that how about reactive oxygen species? What can happen if, for example, in the case of H2O2, I change the proton to deuterium, and what can be the effect on the metabolism of a tumorous in the normal cell? I know that it's not your perfectly subject, but maybe you can answer.
Well, it's part of my subjects, but indeed, and you're right, there's two branches of the pentose cycle. One is what we call the direct glucose oxidation, as the glucose 6-phosphate dehydrogenase dependent process, and that's the one that generates NADPH for reductive synthesis that is involved in fatty acid synthesis. Actually, it's not that particular flux is not very responsive to DDW treatment, and we looked at it as well, because that's also part of, obviously we always measure ribose flux or glucose to ribose flux through the oxidative and non-oxidative branches of the pentose cycle. It seems that because in DDW citrate is in excess, actually NADPH is produced in the cytosol, because NADPH production takes place through citrate, isocitrate. There's this IDH isomerase mutations that are linked with cancer. That's the process. This is the cytosolical soluble isocitrate dehydrogenase. It seems that after DDW treatment, most of the NADPH synthesis or generation process switches to the cytosol. So it seems that the pentose cycle, just to answer specifically your question, is really not a responsive flux to DDW depletion. Now, yet I have to tell you that actually in cancer, in tumor cells, it seems that actually the oxidative branch of the pentose cycle is not that critical for nucleic acid synthesis. It's actually the non-oxidative branch, which in some tumor cells is upregulated and actually produces all the way to 80% of ribose or deoxyribose for nucleic acid synthesis. And even for DNA synthesis, for glutathione reduction, the reducing equivalent comes from cytosolic isocitrate dehydrogenase reaction. So some tumors can really escape from the oxidative branch, because the oxidative branch is controlled by NADP availability. So that's why tumor cells don't really like to use it, because if they do, then they cannot really produce sufficient supplies of nucleotides or nucleic acids, simply because it's an NADP dependent process and it's irreversible. In the meantime, the non-oxidative branch, which is transcromase and transadolase driven process, it's more like just a kind of it's a carbon exchange, set of carbon exchange reactions. There is net ribose synthesis through the non-oxidative branch, and obviously for some tumors and for some tumor phenotypes or certain levels of transformation, actually they start using the non-oxidative pathway pretty efficiently.
Okay, thank you.
Mm-hmm.
Can I just ask a subsidiary question to that, actually? What about turnover of the reduced nucleotide pool? Do you see big changes in that, both in terms of the relation to tumor, but also in terms of the presence of DDW?
So
the turnovers of the NADH and NADPH pools, which are very critical in metabolic regulation.
Yeah, it's really interesting, because you don't have to measure the DNA or deoxyribose turnover pool because your lignosulfate data, because you can't really pack without nuclear membrane. Really, the cell cycle process would not proceed without sufficient supplies of lignosulfuric acid.
Yeah.
So when you correlate those two guys, those are almost just like identical. Those are in this 99%.
It gives you a proxy for that turnover.
Yeah, it gives you a proxy. But yeah, we measured all those correlated, those in various... Yeah, so we measured those in correlation. So I didn't show that data, but that would be similar to lignosulfate data, yet we know it can be used as a marker.
Okay.
Yeah.
Right.
I would like to say only a short remark that I'm very pleased that we are working together and it's very fantastic your results, and I hope that your results we really have that go closer and find the cancer.
I mean, listen, it's not fantastic. It is what it is. The truth is, yeah, I mean, what can I say? I mean, you know, I just kind of, yeah, it's just, it is what it is. However, you have to be aware of that there are clear applications that you can actually measure. You can actually submit as this is your marker for this particular flux. I work with pharmaceutical companies. They love to inhibit fatty acid synthase. They would love to generate this type of molecular crowding, just simply because that's what it is.
I think it's interesting that you transfer your secrets while driving around in cars. This is particularly necessary in the internet age. I think never send anything by email that actually matters. I logged onto my computer yesterday afternoon, having got here a few hours earlier, and lo and behold, it sort of came up and said, "Oh, hi, you're now in Hungary. Here are all these adverts for the sort of things you can do in Budapest." So
well,
you really are traced.
Yeah, well, actually, efforts of developing specific fatty acid synthase inhibitors, it's well published in the medical literature, so I don't tell anybody's secret if I tell you that those efforts are, you can check them. So it's really nothing secretive. Yet, it seems from the efficacy point of view, there might be some really useful comparison type of biomarkers that we can carry from here.
Yeah. Right. So do we have any other questions or comments on this outstanding contribution? If not, thank you again very much indeed for... Would you like to introduce the other speaker?
Yeah.
So our next... since László has been talking for a long time, he would like to give his voice a little rest, so he's asked me if I would also introduce the next speaker, Dr. Nagy from also from Hungary, of course, who's going to talk to us about the effects of deuterium-depleted water as a chemotherapeutic agent for different tumor cells. So, over to you.
Thank you.